Rekin has launched the Brain Minds Data Portal, a resource of brain data of the common marmoset, a primate closely related to humans. A key dataset is the marmoset PFC connectome. The prefrontal cortex, or PFC, plays a central role in orchestrating various cognitive functions, and understanding its architecture in the marmoset is critical in extrapolating to humans. In this study, a viral tracer is injected into the PFC. The tracer infects neurons, causing them to express a fluorescent protein that travels down their axons to their terminal sites. Afterwards, the brain is imaged and digitally reconstructed into 3D, with the tracer pathways highlighted. This is repeated on many subjects to build up a standard brain map of PFC connectivity. We have developed interactive web pages for exploring this data. These are the Tracer Injection Viewer and the Tracer Injection Connection Matrix. In this video we show you how to use these web pages. The Tracer Injection Viewer is a web page that acts as a central hub to explore the data. The Tracer Injection Viewer initially displays a cortical flat map with the injection points of all experiments. The cortical surface view can be optionally displayed too. When the mouse cursor hovers over a region, its name and number of injections received are displayed above the flat map. The table on the left displays all tracer injection experiments. The experiment list can be filtered by region, either by typing a region name or by selecting regions within the region tree view. When hovering over an experiment line in the table, or its injection location on the flat map, a tracer signal preview thumbnail is displayed. The viewer can switch to layer view mode with this button. The tracer signals of top, middle and bottom layers are overlaid on the flat map. Visibility of individual layers, or group, may be toggled on and off. The tracer signal's contrast can also be adjusted. The viewer can switch to sample overlay mode with this button. Up to 6 middle layers from different experiments can be compared. Detailed information about the experiment and the corresponding marmoset subject is available. For each experiment, linked to brain section viewers are provided for exploring original fluorescence images and tracer segmentations for all serial coronal sections. Or the 3-plane version to also explore along axial and sagittal axes. Check out the guided tours that will further explain the viewer and its features. The connection matrices show summaries of interregional connectivity. Each row of the matrix corresponds to a single experiment. It displays the projection strength between the experiment's region of injection and other cortical regions, which are displayed in columns. The connectivity strength is determined by measuring the amount of tracer that migrated from the injection point to other regions of interest. In the matrix, the connectivity intensity is displayed by an easy-to-understand color scale, allowing quick comparisons of relative strength of connectivity within or between experiments. By using the interactive matrix viewer, we can explore the wide range of connection intensities by changing the scaling method or adjusting levels boundaries, and see the update of the matrix in real time. The laminar map connection matrix is a more complex variant of the interactive matrix viewer, which gives insight to the signal depth variability within the flat matrix. Tracer signal expressed at different cortical depths are grouped into top, middle, and bottom, color-coded and overlaid onto the matrix, in a similar fashion to the tracer injection viewer sample overlay mode. The matrix helps users find datasets of interest and then look at them in more detail using the zoomable viewers. By clicking on any matrix cell, a details panel will open with the option to easily load the relevant section viewers. The Zooming Atlas viewer is used to explore the original high-resolution fluorescence image and tracer segmentation for all serial coronal sections of an entire brain. It can be accessed from the tracer injection viewer or connection matrix. Initially, it displays an uncluttered view focused on brain images. Expand the tool panels located on both sides by clicking on their vertical handle. The view can be changed using intuitive mouse controls. Zoom in and out using the mouse wheel. Pan by dragging the image. Panning the current section is also possible by dragging on the global view thumbnail. The currently viewed slice can be changed by clicking on the slice navigation slider, or even on the image below, which displays a generic sagittal brain view, where the current section is shown by a red line. Viewed locations are recorded in the web browser history. It's possible to use the back and forth button, or bookmark a specific view. As in the tracer injection viewer, it's possible to show the current dataset's details. Brain regions are displayed in a hierarchical tree view. Search a region by typing its name. When a region is selected in the tree view, a region detail panel appears at the bottom. It contains a button to directly jump to the center slice of the selected region. Atlas regions are figured by colored areas overlaid on top of the brain image. Selected region is synchronized between image and tree view. The region tree view includes a filter to only show regions that are present in the current slice. Region overlay visibility can be modified. Each image layer visibility can be adjusted independently. Image contrast and gamma can be adjusted to take advantage of the wide dynamic range of 16-bit images. All settings are stored across sessions. A measuring tool allows the measurement of image features. Discover more by taking the guided tours. Primate brain research is essential for understanding the human brain and developing knowledge-based strategies for the diagnosis and treatment of mental and neurological disorders. Through the Brain Minds Data Portal, we aim to promote open data, research, and collaboration for the advancement of neuroscience and the treatment and diagnosis of human neurological disorders.